Have you ever heard about the crowned pigeon? Welcome to your channel ADP for Learning. My name is Ashish. Today, we will explore and learn about the genetic basis of crown-like mystery in pigeons. Before going deep into the topic, utilize a few seconds of yours to like, share, and subscribe to your own channel ADP for Learning. Now coming back to the topic of crowned pigeons or pigeons with a crest. As we have seen in our yesterday's video of why pigeons are an ideal animal for genetic study. The importance of these birds in genetic research. We have learned that some pigeons have smooth heads like their wild ancestors had. But on some domestic pigeons, the feathers on the back of the head and neck stand up to form a crest like a crown. Both of the pigeons shown in the picture are colored spread blue. The spread allele, or one of the variants of genes located in the genome of pigeons, causes the color to be evenly distributed across the body. As shown in the figure, the one pigeon has a small crest, while the one on the left has no crest. If you closely observe the pigeons both phenotypically and genotypically, then you will be surprised to find out that only a single gene controls the presence or absence of a crest. This gene comes in two different versions, or alleles, namely crest and no crest, or in other words, the wild-type pigeons don't have a crest while the non-wild-type pigeons have crest. If you are confused with the term allele, you can check my previous video named "What are traits?" on your own learning channel ADP for Learning. Now again, coming back to the topic after a little self-promotion, the figure represents that the pigeons inherit two copies of the crest gene, one from each parent. The two alleles together make up a bird's genotype. What we see, also called the phenotype, is the physical outcome of these two alleles. The no crest allele is dominant to the crest allele. To have a crest, a bird must have two copies of the crest allele. If you dig deep for finding out the molecular basis of the crest formation in the pigeons, then you will see that inherited characteristics are the products of proteins, and proteins are coded for by genes. Ultimately, the differences in humans and pigeons' inherited characteristics arise from differences in the DNA sequences of human and pigeons' genes. DNA differences can affect how protein functions, or when and where a protein is made. In 2013, a group of researchers at the University of Utah, led by Professor Mike Shapiro, identified the genetic variation that causes the crest phenotype. To find the crest gene, the research team looked at the genomes of 42 pigeons, which includes 37 breeds, plus 2 feral birds and a closely related species, some with crests and some without. They compared the DNA sequences, and they found a region of DNA that was different between crested and non-crested birds. The region contained one gene, called epherin receptor B2, or also known as FB2. When the researchers looked at another 130 birds, they saw a 100% correlation. All birds with crests had 2 copies of the crest version of FB2, and all birds without crests had at least 1 copy, and more often 2, of the no-crest version of FB2. Researchers also find out that groups of 3 nucleotides along a stretch of DNA code for specific amino acids in a protein. The protein coding portions of the crest and no-crest alleles differ by just 1 nucleotide, and they code for proteins that differ by just 1 amino acid. A small portion of the change in the DNA and amino acid sequences is shown in the figure, where the wild-type no-crest pigeon protein consists of the amino acid arginine, while the pigeon with crest has cysteine amino acid instead of arginine. So, the question may arise, why do the crest and no-crest pigeons allele code for slightly different proteins? The answer to this question lies in the fact that the crest phenotype is visible very early, while a chick is still developing inside the egg. As soon as neck and head feathers appear, they start to grow backward. Instead of pointing down toward the tail, the feathers point upward toward the top of the head. The PHB2 protein appears to have something to do with setting up feather orientation even earlier in development. On the other hand, the no-crest allele codes for a protein that functions normally. As shown in the figure, the normal protein sits in the cell membrane, and when a certain signal from outside the cell attaches to it, it transmits a signal to the inside of the cell. The crest allele codes for a protein that is broken. The 1 amino acid difference changes the shape of the protein, making it unable to transmit the signal. A bird with 2 normal alleles makes only normal EPHB2 protein. When a bird has 1 normal that is no crest and 1 broken that is crest allele, its cells make both normal and broken protein. There still is enough of the functioning protein around to transmit the signal. But a bird with 2 broken alleles makes only broken EPHB2 protein, which can't transmit the signal as shown in the figure. We don't understand exactly how, but when the signal can't get through, the neck feathers grow upside down, and the bird has a crest. So, at last, the question may arise, what are other genes that influence crest size and shape? As we have learned that one gene controls whether a pigeon has a crest or not, similarly a variety of other genes influence crest size and shape. These genes are often called modifier genes. You can think of them as genes that modify the basic crest phenotype or physical appearance. All of the birds shown in the figure carry 2 copies of the crest allele. Some of the crests are subtle, and others are quite extreme. The differences are due to different modifier alleles or gene variants. There are probably several modifiers, each with slightly different effects. These genes were likely selected by multiple breeders who were working with different breeds. We will learn about these modifier genes in our future videos regarding how exactly they affect crests or interact with EPHB2 or other proteins in the same molecular pathway. Thank you for joining me on the amazing journey of learning. We will again meet with new innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment. Your feedback will act as a valuable tool in making future videos better.